So the first part of photosynthesis are the light-dependent reactions. So remember, these occur in the thylakoid membranes of the chloroplast. So if we look at the bottom left picture here, here's the chloroplast. And remember, the thylakoids are these little green discs inside the chloroplast. And so in the membrane of the thylakoid, that's where the light-dependent reactions are going to occur. And the whole purpose of light-dependent reactions is we're going to be capturing that light energy and then using that to produce ATP and NADPH. And in order to capture the light energy, we're going to use photosystems. And these photosystems are what is embedded in the thylakoid membrane. And so in this middle picture, you can see one of these photosystems. And embedded in these photosystems are the chlorophyll molecules that actually absorb the light energy. And so they're going to be passing around the light energy, and then they finally get to the center here, and that's where they're going to boost the electrons up and then send them down an electron transport chain, which we'll see in another picture here. And the electrons that are going to be lost from photosystem 2, which is the first photosystem that's going to be used here, we need to replace those electrons. So where we're going to get electrons from is from water. And so we're going to split the water and rip the electrons off water to give them to photosystem 2. And then what's left will be oxygen. And so oxygen will be a byproduct. And so oxygen is actually going to be released from these little holes that can open and close on the underneath of the leaves on plants. And so if you have an underwater plant, you can actually see the oxygen bubbles coming off underneath the leaves, which is pretty cool. So you can actually see that in this picture here. Right, and as the electrons pass down the first electron transport chain, those electrons are going to release some energy, and that energy is going to be used to actively transport hydrogen ions from the stroma, so this fluid outside the thylakoids, into the thylakoid space. So there's a space inside these little green discs inside the thylakoids. So you can see the little dots here, those are representing the hydrogen ions. And so there's less hydrogen ions outside here in the stroma, and there's a lot more hydrogen ions inside the thylakoids in this thylakoid space. So we're going to be moving the hydrogen ions from low concentration to high concentration inside the thylakoid space. So going from low to high concentration, that would be going up the gradient, so that would be active transport. And so to do that active transport, we're just going to use the energy from the electrons. And then the hydrogen ions will diffuse down their concentration gradient from the thylakoid space back out into the stroma, going out through ATP synthase. And as they go down their concentration gradient, they're going to release energy, and that energy will be used to spin the ATP synthase, and that will be used to make ATP. So let's take a look at a picture here. So this is just showing you a thylakoid. And the stroma is all around the thylakoid here. And then inside here is the thylakoid space. And you can see these two photosystems embedded in the thylakoid membrane. And here's photosystem 2. So light energy will hit photosystem 2. We'll use that light energy to boost the electrons up, send it down our first electron transport chain to the next photosystem. And then as the electrons go down this first electron transport chain, they're going to release some energy, and we'll use that energy to pump hydrogen ions. And so we're going to take the hydrogen ions from a low to a high concentration, And so that's active transport, and the energy from the electrons is going to be used to do that work. And then we're also going to need to replace these electrons that we lost. And so we're just going to split the water apart, take the electrons off the water, and then we're left with oxygen as a byproduct. And then the electrons that we get from water will eventually end up here on NADPH, our electron carriers. So we boost them up again with more light energy in photosystem 1, and then they go down a second electron transport chain and end up on our electron carrier NADPH. Okay, and then these hydrogen ions that are inside the thylakoid space will then diffuse down their concentration gradient, going through ATP synthase. They'll release energy, cause the ATP synthase to spin, and that will give us the energy to make ATP. So you can see the two things that are made in the light-dependent reactions will be ATP and NADPH. And then the ATP and NADPH will be used by the Calvin cycle to make the carbohydrate or sugar. Okay, so the Calvin cycle is going to occur in the stroma of the chloroplast, and it's going to use the ATP and NADPH from the light-dependent reactions to make a carbohydrate. Okay, so we're going to start with the reactants. So we've got ATP and NADPH, which came from the light-dependent reactions, and then we're going to get carbon dioxide gas from the air. And then it ends with the products that are made, this 3-carbon sugar called glyceraldehyde 3-phosphate, which is a mouthful. So a lot of times we just abbreviate that G3P. And then we also end up with ADP and NADPH+, okay, because we've used the energy from ATP and we use the electrons from NADPH. And so this is our empty electron carrier now. We've dumped the electrons off. So, and this is our full electron carrier over here. Okay, our full dump truck. Okay, and the other thing in the Calvin cycle, because we are going to be using carbon dioxide gas from the air to actually make food, to make the carbohydrate or the sugar, and so we need an enzyme to help us do that. And so this really unique enzyme is called Rubisco. Okay, and Rubisco's job is to fix carbon dioxide. So that just means that it's able to take carbon dioxide and make it part of an organic molecule. And the important thing to remember about this enzyme Rubisco is that it's probably the most abundant and most important enzyme on Earth, because we are not able to do anything with carbon dioxide. To us, it's just a waste product. We just exhale it. We can't do anything with carbon dioxide. We can't make anything with it. But plants and algae and cyanobacteria, those photosynthetic organisms, because they have this enzyme Rubisco, they're able to take carbon dioxide gas and they can make things with it. So they can make a carbohydrate, and then from there they can make other biomolecules, so like proteins and lipids and nucleic acids. So because of photosynthesis, this is where we end up getting all of our carbon from, because we eat the plant or we eat the animal that eats the plant. So, okay, and this is just a reminder where the Calvin cycle is occurring in the chloroplast. So Calvin cycle is out in the stroma, this fluid part outside the thylakoids. And we're just going to be taking carbon dioxide, fixing it, making it part of an organic molecule, and then we'll make our carbohydrate or sugar, and we need the energy of ATP and NADPH from the light reactions to be able to do the Calvin cycle. Okay, and this is kind of a scary picture, but it's just showing you that with the Calvin cycle, we're just going to take carbon dioxide and with the enzyme Rubisco, which is going to fix the carbon dioxide and make it part of an organic molecule. And from that, we can make our G3P, our glyceraldehyde 3-phosphate, and we can make our carbohydrate or sugar from that. And then this is also important to know. So for every 6 glyceraldehyde, 3 phosphate molecules or G3P molecules that are made by the Calvin cycle, one of those G3P molecules will be used to make sugars and other organic molecules. And then the rest just stay in the Calvin cycle to remake some of these intermediate molecules that are used in the Calvin cycle.